At the Lockwood Lab, we've taken theory from traditional aquatic environmental DNA sampling methods and pioneered new methods in order to study terrestrial organisms. Just as aquatic life will release DNA into the environment, terrestrial organisms will do the same in the form of exoskeletons, excrement, mucus, or skin cells. The DNA they leave behind is what we call environmental DNA, or eDNA. Having the ability to collect eDNA samples on land gives us the opportunity to efficiently assess the biodiversity of terrestrial ecosystems or assess the presence of a particular species in that environment. The molecular methods we use to assess environmental biodiversity are so sensitive that we only need to pick up trace amounts of DNA in order to detect an organism. Over the course of this video, we'd like to demonstrate one of the primary methods we use to capture terrestrial eDNA off of tree surfaces. You'll see our method uses self-desiccating filter housings, but other commercially available and reusable filter housings can be used. Before we begin, we want to emphasize some standard practices that we follow and the importance of avoiding contamination when collecting samples. eDNA sampling is a very sensitive tool, so minute traces of contamination can cause false positive detections of an organism. To combat this, we thoroughly clean all of our sampling equipment with a 10% bleach solution, wear clean gloves, and change them often, between samples and whenever there is potential contamination occurring. We avoid touching any equipment that may come into contact with a sample, and we make use of negative control samples to detect any potential contamination. We typically collect field negatives using the same equipment used for sampling, but in the place of a field sample, we use deionized water. Ideally, a negative control is collected before beginning sampling at each field site, but minimally, field negative controls should be collected at the beginning and the end of each sampling day. As we go through our sampling process, you'll get a chance to see just how carefully we handle our sampling equipment. If interested, you can find a list of all the equipment we use for this particular method on the Rutgers eDNA Lab website. To begin sampling, you'll want to put on a fresh pair of gloves and set up your peristaltic pump and tubing. Be sure to keep the front end of your tubing clean throughout the sampling process to avoid any unwanted contamination. The back end of your tubing should be placed into a graduated cylinder in order to measure the amount of sample filtered, which can be an important variable for later analysis. Next, you'll want to assemble your sampling pole with a new clean pair of gloves, paying close attention not to touch the metal of the roller handle or the damp paint roller itself. The sterile bag that the pre-clean roller is stored in can be used to handle the roller when placing it onto the pole. Once assembled, you'll begin rolling the surface of the target tree. We typically roll from the base of the tree to about 3 meters high, making sure to also roll the tops of any branches within that range. Some things to keep in mind. The behavior and life history of your species should be considered, such as where it feeds or spends most of its time. Targeting a specific portion of a tree during a certain time of the year can mean the difference between detection or non-detection of your target organism. The number of trees you'll want to roll to generate a sample will depend on the abundance of your target organism, the spatial extent of the study area, and your budget. We recommend rolling between 3 and 6 trees per site. Recent weather conditions should also be considered, as rain, in particular heavy rain, can wash away surface DNA. After the target trees have been rolled, you'll want to carefully place your roller into a sterile bag and fill the bag with about 150 milliliters of clean DI water. In this case, we're using a pressure sprayer, although a carboy can also be used. The roller then gets massaged and shaken for about 30 seconds to release the DNA from the roller and suspend it in the water. The roller is then pushed out of the sterile bag to allow the water to be filtered. At this point, you can grab a clean filter. Typically, we pre-label the bag with the site name, date, and sample number. Prepare the filter housing with a fresh pair of gloves. Carefully attach the bottom of the filter housing to the pump tubing by using the filter bag to manipulate the filter. You'll then want to attach the snorkel provided to the bottom of the filter housing. Once your filter is prepped, insert your filter snorkel into the sample bag. The pump will begin to move your sample through the filter housing, capturing your eDNA field sample inside. Once all the water is filtered, you'll want to remove the snorkel from the bag and invert the housing. You can then remove the snorkel from the housing, slightly increase the speed of your pump, and let the pump continue running for another 30 seconds to allow your sample to dry. Detach the tubing from the back of the filter housing and place the housing in the labeled bag for storage. Be sure to remove as much air as possible before sealing the bag. The sample is now ready for transport in a cool, dark container, such as a cardboard box or cooler. While our lab primarily uses self-desiccating filters for this method, you can use reusable filter housings. These assemblies should be thoroughly cleaned before use and assembled in a control setting whenever possible. Because they're not self-preserving, filters from reusable housings should be pulled as soon as possible and be kept cold or preserved in non-denatured ethanol to prevent degradation of the sample from occurring. As we mentioned earlier, it's important to take negative controls before you begin sampling at a given site. This can be done first thing at the site after assembling your sampling pole. Immediately remove the roller with a sterile bag and proceed with the steps as mentioned earlier to collect a field sample. Collecting a negative control at the end of the sampling day is also useful to ensure the sampling equipment did not carry over target DNA between sites. This method has been utilized by our lab to detect invasive insects on the surfaces of trees, but can be adapted and utilized for a variety of different surfaces. For example, we've used this method to detect the presence of little brown skinks under cover boards and to classify which bat species are roosting under local bridges. From the Rutgers eDNA lab and our partners, we hope you found this helpful. For more information on our sampling methods and eDNA in general, please visit our website using the QR code provided. Happy sampling!